Many G protein-coupled receptors have a large extracellular ligand-binding domain. When an appropriate protein ligand binds to this domain, the receptor undergoes a conformational change that is transmitted to its cytosolic regions, which now activate a trimeric GTP binding protein, or G protein for short. As the name implies, a trimeric G protein is composed of 3 protein subunits called alpha, beta, and gamma. Both the alpha and gamma subunits have covalently attached lipid tails that help anchor the G protein in the plasma membrane. In the absence of a signal, the alpha subunit has a GDP bound, and the G protein is inactive. In some cases, the inactive G protein is associated with the inactive receptor, while in other cases, as shown here, it only binds after the receptor is activated. In either case, an activated receptor induces a conformational change in the alpha subunit, causing the GDP to dissociate. GTP, which is abundant in the cytosol, can now readily bind in place of the GDP. GTP binding causes a further conformational change in the G protein, activating both the alpha subunit and beta-gamma complex. In some cases, as shown here, the activated alpha subunit dissociates from the activated beta-gamma complex, whereas in other cases, the 2 activated components stay together. In either case, both of the activated components can now regulate the activity of target proteins.
Adenylyl cyclase is a membrane-bound enzyme whose catalytic domain is activated by the GTP-bound form of the stimulatory G protein alpha subunit, or G-alpha-S for short. Activated adenylyl cyclase converts ATP to cyclic AMP, which then acts as a second messenger that relays the signal from the G protein-coupled receptor to other components in the cell. In most animal cells, cyclic AMP activates cyclic AMP-dependent protein kinase, or pKa. In the inactive state, pKa consists of a complex of 2 catalytic subunits and 2 regulatory subunits. The binding of cyclic AMP to the regulatory subunits alters their conformation and liberates the catalytic subunits, which are now active, and phosphorylates specific target proteins. In some endocrine cells, for example, the activated pKa catalytic subunits enter the nucleus, where they phosphorylate a transcription factor called CREB. Phosphorylated CREB then recruits a CREB binding protein. This complex activates transcription after binding to specific regulatory regions that are present in the promoters of appropriate target genes.